everyone. Oh, there's no one here. Well, we're still going to continue the conversation anyways, right? So, we had left off last class talking about different types of ways that we can get proteins into different locations inside the cell. So again, let's go back. Let's remember this, right? So, ways to have proteins, not membranes. We said in total there were 3. So the first one we have, right, is crossing across into the nuclear pore. The second way that we said was that we want membrane proteins to actually cross the membrane. So, crossing over membranes. And we said that there was a total of 3 different ways that we could actually get proteins across membranes. The first is sometimes we would actually just need to deliver them into the inner lumen or into the inner matrix of the cell. We said that those are soluble ones, right? We also may actually need to have our proteins stay stationary in the membrane, and those would be something that we call our single pass. The last and final one where we left off last week, right, was the double pass. And so again, if we look, right, the single pass, we still have our N-terminus, and what we're looking for now is the fact that we have this hydrophobic stop transfer sequence, and what's going to happen is as it comes through our protein translocator, right, our N-terminus sequence is still going to be present. It's going to keep kind of a hold position inside the membrane as a whole. And as that hydrophobic start transfer sequence enters now into the protein translocator, our signal peptase actually receives that information, cleaves off the molecule, right, cleaves off our N-terminus molecule, and it cleaves off the rest of the protein. The protein officially unwinds, and now we have a single pass in the membrane. Additionally, we said, and this is where we left off last time, right, we said that we could also have a double pass protein. So again, we said that this would be a very unique way to test this, right? We kind of did some experimental research last time we were in class. And we could test this by moving maybe either the hydrophobic start transfer sequence, we could even move the N-terminus sequence, right? We could cleave off our protein peptidase, right? But importantly, these are all ways that we can get very important proteins either across membranes or physically embedded into membranes, right? Because that's very important. The cell has to constantly maintain all of the different proteins that are able to do work, including our protein translocators, right? So, another way, and the third and final way that we can actually have proteins cross into different components in different parts of the cells is maybe we actually distribute our proteins through transport vesicles. Right? And again, these are different types of transport vehicles, if you will, right, that are going to carry soluble proteins and our membrane proteins, right, to their correct destination. So if we look at this, right, we have the nuclear envelope. We've already discussed how do we actually get things into the nuclear pore. We now are going to take and focus a little bit away from distributing things necessarily inside of membranes. We're talking about how do we package things and appropriately ship them off to correct parts of the cell. So we already know that the Golgi apparatus is sort of our UPS station inside of our cells. And again, they are the kind of the final packaging place in which proteins that maybe were just processed in the ER, or maybe even cytosolic proteins that were packaged, they might need to be incorporated into vesicles, right, and transported to other locations within inside the cell. So we can have transport vesicles that will bud off of the Golgi apparatus. Maybe sometimes their final destination is to deliver proteins outside of the cell, right? We'll talk a little bit more about this in chapter 16 when we talk about signaling molecules. Or we may very well need to bring things in from the outside, right? We may have to actually endosize different types of materials and bring them into the cell because we may need to use those for further signaling cascades. Again, we're going to come back to that in chapter 16. So, how does this process occur? Let's grab an eraser. This is a very nice whiteboard, guys. I know, right? Very nice whiteboard, everybody. All right, so vesicle budding, right? This is one way that we can actually internalize things into the inside of the cell. So if we look at this EM picture, right, what do we see? From A, right, we actually see that parts of the membrane are starting to pocket inward, right? So we are actually trying to bring in, whether it be different proteins, whether it be food molecules, whether it be water, right? Large amounts of water, right? Instead of if we're trying to bypass through our aquaporins, maybe the cell needs a massive amount of water. Sometimes we will actually form an endocytic vessel to do that. So if we look and take time points all along this, right, we can see that the membrane infolds, but more importantly, the membrane seals up around behind it. Again, this is very important. We don't want to keep our membrane open and exposed for very long. If we look a little bit deeper, right, if we take and kind of zoom in on one specific part of this process, what we're actually looking here is what's known as clathrin-mediated endocytosis. Let's write that down. Now, the breakdown of this terminology just really means that clathrin, right? Clathrin is just serving as the coat mechanism. So if we see all the way over here in kind of the first stages, right? We have some receptor molecules. These receptor molecules or these receptor proteins are going to accept individual receptor cargo, right? And now that signaling cascade says we need to internalize whatever this cargo is that is now bound to this receptor. In order to do that, right, we start to bulge the membrane inward. But how do we do that? Well, we actually form a coat or a cage-like structure, if you will, all around the actual membrane. That helps then to actually cause pressure, right, to deform the membrane and actually default and bring the membrane inward and eventually pinch off to release that cargo inside the cell. So let's walk through this. First thing we have, right? We have our cargo receptor. That binds to some particular signaling molecule. Right? What this does then is it induces the pathway of clathrin-mediated endocytosis. And to make this easy, because that's a big long word, let's just call this a CME pathway, right? So as we bind our specific cargo to our receptor molecule, this triggers right? And what do we mean by it triggers the CME pathway? It recruits all of these different components, right? All of these different molecules to the actual inside surface of the plasma membrane, right? Because again, here we are outside the plasma membrane and the cytosol being the inside of the plasma membrane, right? Once our cargo molecules bind to our cargo receptor and trigger that activation pathway, that that signaling cascade that we'll talk a little bit more about in the next chapter signals to recruit clathrin coat, signals to recruit the adaptin molecule, right? And again, so on and so forth. It keeps recruiting these things until it eventually starts to pull the membrane inward, right? So as we can see, it's going to keep pulling that membrane inward. And actually almost forms, well, if you look at it, it almost kind of forms the reunion tower ball, if you will. But the idea here is it forms this inner puckering, or this infurrowing of the actual plasma membrane, surrounded by our cage-like structure of our clathrin coat. And once we have pulled in, this process is called invagination. Once we pull this in, we have to pinch it off of the membrane, right? So this triggers the clathrin-mediated pathway. CME will continue. And again, it's going to continue to do this until a full cage-like structure, that clathrin coat, is about 3/4 surrounding its cargo, right? It's got a nice good grip, I should say, on the actual cargo itself. Once that happens, once we have pulled in the majority of the cargo that we need, dynamin is then recruited. And what this does is it's literally a twisting mechanism that continues to constrict around and around and around and around the neck until eventually it pops off the actual entire endocytic vesicle, right? So dynamin is recruited. And again, dynamin will do its job of constricting, right? It'll continue to twist, and each time that it's closing and wrapping around, right, it's constricting the neck of that actual vesicle until it pops the vesicle off of the membrane reseals behind it, right? And what we have is now our internalized cargo. The very, very final step is the clathrin coat disassembles. And again, now we have brought in our cargo transport molecules, right? We now have our cargo inside of the actual cell. So how does this really, truly happen, right? We kind of got a first taste of this, right? But there are so many other mechanisms that are actually behind this, and we're going to touch briefly upon this.